Thank you very much for the invitation. It's wonderful to talk about some of our recent work on the physics of how protein diffusion can control how cells respond to electric fields. So I'll give you the basic introduction to my group. We work on the physics of cell motility using theory and computation, mostly with eukaryotic cells, so not quite like the bacterial cells that you were just seeing. So why do we care about cell motility? And of course, at a biological level, that answer might be thinking about what happens when you get a cut. So let me show you the classic movie of this, wonderful white blood cell managing to chase down a bacterium. And so obviously, you need motility of these individual cells for this to function. This is just taken in a slide of blood, but this is happening, of course, within our body. And you also need motility for things like wound healing, where here a layer of epithelial cells is invading into this new region where there was a circular region cut out. So that's sort of like, the reason why you might care about this as a human being, but specifically as a physicist, I think cell motility is this beautiful question, both because you see like that dynamic change of shape and flowing of the cell. So cells are a great soft matter question. But I'm not going to talk so much about that today. I'm going to talk about a different element, which is that, of course, cells, in order to respond to something like a bacterial cue, need to be able to sense their environment. And this is often a very stochastic process. For instance, like when sensing bacteria, cells can sense things at a nanomolar concentration, so somewhere on the order of a molecule per cubic micron. So cells often have to make sense of highly stochastic signals. And so what I'll talk about today is what that means for physical limits of sensing electric fields. And this is work primarily by Iffy, a graduate student in my group who just defended late last year. Okay, so why should cells sense electric fields? Here I'm thinking of eukaryotic mammalian cells especially. So in particular, this is important in wound healing. And that's because if you have a region of tissue that's injured, there's an electric field that points towards the wound. And this happens because in normal healthy tissue, there's a potential difference between different layers of skin. But if there's a wound, that basically short circuits that potential difference. So you can see that there's a potential difference between healthy skin and damaged skin, leading to an electric field pointing towards the wound. And that's part of the cue that cells follow in order to ensure that they are traveling towards the wound when they're healing or if immune cells are trying to get to a wound. And you can recapitulate this by applying a directed DC electric field. This is on a keratocyte, so this is a scale cell for a fish. And you can see that it manages to turn the cell and cause it to move in the direction of the electric field. So how does a cell actually respond to a field like that? So let me give you the sketch of this first, and then we'll deal with the molecular details later on. There are charged molecules on the surface of the cell, basically membrane proteins, that we'll call sensors. And I'll tell you a little bit later on what the sensors actually are. And if you apply an electric field, these sensors become redistributed because they're charged. So a combination of electrophoresis and other effects will lead them to redistribute moving around the surface of the membrane. Then there's an asymmetry from one side of the cell to the other, and then that can lead to the cell polarizing and moving in that direction. So if I tell you this, what physics would then limit cell accuracy in an electric field? So here are two simulations of spherical cells. Each of these red dots is the location of a sensor that's undergoing some random diffusion. So diffusion is mixing it up, but then they're charged, so they're being pulled to one side. So I want you to think about this. If you're a cell and you're like, okay, where is the electric field pointing? Is there even an electric field that is showing up in this case, And I think the answer is it's not obvious if there's an electric field here or not. But, so think about which cell you think has an electric field on it. Now I'll turn the field strength up. So you can see that the field on the left had a field on it, and now it's relatively clear what direction the cell should be traveling. So we think of this as being reflecting this difference in noise from like the randomness of the sensor positions compared with the bias arising from the electric field. So we try to put together a basic model of this point. We describe sensors moving around on the surface of a cell, each with an orientation theta. There are two things happening to these sensors. One is that they can be pulled around by the electric field via electrophoresis. So we assume that their velocity is proportional to the electric field in the plane of the membrane. All of the details of charge and stuff like that get thrown into this one coefficient, which will fit two experiments. And so the other thing that's happening is diffusion. So diffusion is going to try to spread out the sensors. So to figure out the distribution of sensors, the probability of being at a location theta, we balance the flux coming from transport, which is this V times P, with the flux from diffusion, which just tends to spread everything out. If you take care of the geometry, you find this equation, which you can easily solve to find that the probability of having a sensor at a given angle theta goes like e to this factor kappa times the cosine of the angle. Kappa, you'll recognize here, is basically just a Peclet number. It reflects the relative strength of the transport due to the electric field and the diffusion. So like if you are at low kappa, that's diffusion very large relative to transport due to the electric field, you get something where diffusion dominates and everything is homogeneous. If you're at high kappa, then the electric field is much more important and the sensors are strongly polarized to one side of the cell. So we take the perspective of being the cell ourselves and say, what's the best guess we can make about the direction the field is in? And so we use this maximum likelihood approach where we say, okay, we know the distribution of where the sensors are, and that's one sensor's distribution P of theta. So the probability of observing a particular configuration of these sensors is just the product of P of theta if they're independent. And we ask basically, suppose that you sweep through all of the different possible directions the field could be in. We ask, what's the direction that maximizes the likelihood or the probability of observing the particular configuration of angles that you actually get? And we do this both out of just sort of a theoretical interest, but also because we don't actually think cells can do maximum likelihood estimation. But the answer that you get out of this gives you something very simple and we think quite easy to biologically motivate. So we say that if the sensors are at locations theta 1 through theta 4, you should draw vectors to each of those sensor locations and then just add up the vectors, and that's the direction the cell thinks the field should be in. So obviously, if there are more sensors on one side, the cell is going to go towards that direction. Okay, so that's our argument for what the cell should be doing. But there's a limit to this. We said that these sensors are undergoing diffusion. There's some randomness in their location. And we want to ask how accurately can you actually measure the field orientation given all of that randomness? This is limited by a statistical bound called the Kramer-Rao bound. One way of writing it that we figured out is about the directionality. So we'll write this directionality a lot. The directionality is the cosine of the angle between the direction the cell is actually going, the cell's estimate of the electric field direction, and the true field direction, averaged. So if the directionality is +1, that means that the difference in angles is 0, and the cells are always going in the correct direction. If the directionality is 0, it means that the cells are randomly oriented. Okay. This directionality is bounded by something related to a Fisher information. The information reflects the amount of knowledge that the cell could possibly have about the field direction. And it's something that you can compute relatively easily if you know that probability distribution. What we find here is that the Fisher information is really simple. It's just proportional to the number of sensors and the Peclet number squared. So that means that you have more ability to sense the field if the field is stronger, or if you have more sensors, or if you have a larger cell radius. And that all makes sense. But there's a more important prediction that comes out of this, which is that we would predict that the directionality should really only depend on the electric field through one characteristic value. And that's this characteristic electric field, and would have this sort of universal form. So what we did was we went to the experimental literature and we found measurements for directionality as a function of electric field for different cell types. So here's for granulocytes. And what we have to do is we have to then fit the characteristic electric field for each of these cell types. And for granulocytes, we see that it collapses okay onto this curve. For keratocytes, those fish scale cells that I showed you before, it also looks like it collapses okay onto the curve. And then for neural crest cells, you see again, we can collapse it roughly onto this curve. And this is not a perfect collapse. I would say this is suggestive rather than conclusive. So we wanted to take this result and sort of push it and see if we could get stronger predictions. And the most obvious thing that we could try to do is to predict how cell directionality changes as a function of cell size. We said that larger cells have more information just because the difference in potential between the back and the front of these cells is larger. And so we said larger cells should be more accurate. We went out to the literature again. And if you go out to the literature again, you saw this wonderful paper, and it has this headline that says, directional sensing does not depend on cell sizes. And so this is obviously a little bit discouraging. And we went to see actually what the underlying data was. So if you replot that underlying data, it's a measurement of directionality as a function of cell size. Each of these points is one trajectory of a cell. And indeed, you look at this graph and you're like, no, there's absolutely no trend here. You can bin it into small cells and large cells, and there's still no significant trend there. And then we asked, okay, well, given the theory that we worked out, what would we predict? We can predict that with the parameters that we fit in the last half. And we'd say, oh, actually, we don't expect a very large change in directionality as a function of cell size. Then certainly not large enough to rule out this model. The reason is largely just that the cell areas are not varying that much from cell to cell. So it's relatively difficult to really get a large effect. So if you do a back of the envelope estimate, they did about 20 cells in this experiment. You need an order of magnitude or more cells to really detect any size dependence. So that's something where we're actually working now with Daniel Cohen at Princeton to try to come up with ways to do these experiments at really high throughput. Okay, so all of that work is published. So now let me tell you a little bit about some stuff that's like unpublished that's I'm really excited about. And that's trying to answer this question of how quickly does a cell respond to an electric field? So if you take an experiment like this one, where you start off with no field, then you turn the field on, and then you see that the cell responds, and you track the cell, then you repeat those experiments like 50 or 100 times, you get a plot like this one. So this is directionality, the cosine of the angle between the cell's velocity and the field direction. And you see that when the field is off, that's this red line you should be reading here, the directionality is about 0. And then when the field turns on, you see that the directionality turns up and then comes to some steady state. Okay, you see that the response time is sort of on the order of 7 minutes or so. And people have done this experiment in other cell types as well. For fibroblasts, you actually see like a sudden rapid jump, and they claim that this leads to a response time of on the order of 1 minute. And so we want to say, okay, what controls the response dynamics if you believe sort of the central assumptions of our model? And the model essentially says that the thing that controls this should be how proteins are moving around on the surface of the cell. And so we now have to write a diffusion equation for that. So we write a diffusion equation for motion on the surface of the cell, and then include the electrophoresis and electroosmotic effects here with the same model as before. And you can take a look at this equation, and if you multiply through by r squared over d, you can see that you can scale out all of the parameters except for one time scale, which is the time to forget, and this parameter kappa. So this time scale of the problem is r squared over d. So d is the diffusion coefficient of the protein. So r squared over d is just basically the time to diffuse the size of the cell. So this is a very relevant number in this problem. So order of magnitude, what do we expect? For typical eukaryotic cells, radii could be somewhere on the order of 5 to 10 microns. Diffusion coefficients of membrane proteins could be something like 0.1 to 0.5 microns squared per second. You get a number of about 1 to 10 minutes. And that looks like we're basically getting the right number, but there's a problem. Fibroblasts are actually pretty large cells, and they're responding in a minute. If you look at these 3T3 fibroblasts, that scale bar I'm showing you is 100 microns. And okay, they're very elongated cells. So if you do an estimate for like sort of an effective radius, you might believe 30 microns, which means that the response time should be way slower, something like 30 minutes. So we went back to our theory and said, okay, what can we do to really quantitatively understand this? And we have a nice quantitative theory for describing how this works. You start with this diffusion equation for the probability of a sensor location P of theta, and we do a perturbation approximation. We assume that the sensors are nearly uniformly distributed, and then there's some anisotropy, A of t, which could be different between the back and the front. So this is saying that A of t corresponds to basically the difference between the back and the front of the cell. And if you plug this in, you see that you can get an equation for A of t, and the only time scale that shows up is that same forgetting time. So it tells you that the response time of the anisotropy in this sensor is just always the time to forget. So it's r squared over d. There's nothing that you can do to avoid that. So does this mean that our theory is just completely wrong? Well, we went through and checked our theory by developing these stochastic simulations. You have these red dots here, and those red dots are the sensors. They're diffusing, and then they're also being biased towards the right-hand side. And then that blue line that's jumping around is the direction that the cell thinks the field is in. So that's fluctuating around a lot. What we plot here is the anisotropy. Again, this is the difference in sensor between one side of the cell and the other, scaled by this Peclet number kappa. And you see that as you turn on the field, that the anisotropy kicks in over a time scale of about the time to forget. But this is a linear theory. So if you multiply the electric field strength by a factor of 5, and then divide that factor of 5 out by dividing by the Peclet number, you don't expect a change. And that's exactly what we see. So if you increase the field by a factor of 5, you still see like the same response. You see the same response if you increase it by a factor of 5 again. So always the sensor anisotropy has this response time of basically the time to forget. However, this isn't actually the direction the cell is traveling. That is given by this directionality, which you can get by summing up all of the vectors pointing to all of the sensors. So this is the cosine of the angle. This is what the experimentalists actually measure is directionality. And you see that when you turn on the field, the directionality comes up. But now if you increase the field strength, you start to see that the directionality starts to respond a lot faster. You crank up the field strength even more, and the directionality's response is faster and faster. So the directionality has a different time scale. And that's fundamentally because this directionality is a nonlinear function of the anisotropy. And if the directionality is a nonlinear function of the anisotropy, the time scale doesn't have to be the same. So what we've done here is we can relate the directionality, which is what experimentalists actually measure, to the underlying sensor redistribution, which is what we think physically is important. So we can try then to start analyzing experimental data in order to address that. So here's an experimental measurement. This is, again, the same experiment from Julie Theriot's group of measuring keratocytes. And then this blue line is the first curve that I showed you. And this is an experiment where they turn on the electric field, and they do this experiment in normal media. And then they did a different experiment, which is, what if they take the same cell type, but instead of growing them in normal media, they put them in a high viscosity media just by adding some polymers to make the fluid more viscous. What they see is that it takes longer for the cell to respond and to turn around. But when I first saw this data, I really was like, this seems impossible. You increased the viscosity of the environment by a factor of 50, but things actually didn't slow down that much. They slowed down by about a factor of 2. But this theory tells us why that has to be. If the time to respond is just related to the diffusion coefficient of the sensor, well, the diffusion coefficient of the sensor, it's a membrane protein. It's kBt over the viscosity of the membrane, but it depends strongly on the membrane viscosity and only relatively weakly on the medium viscosity, simply because the membrane is much more viscous than the water outside the cell. So there's only a weak dependence on the viscosity of the medium. So even if you increase the fluid outside the cell by a factor of 1,000, you're only going to slow things down by a factor of 8. Okay. So now we get to the sort of newer and newer stuff, which is, what is this sensor anyway? And if you'd asked me this question in July, I would have had no answer for you whatsoever. But in September, Nathan Beliveau and Julie Theriot posted this wonderful preprint on bioRxiv, where they did a systematic screen with CRISPR to knock out a lot of potential proteins, and they identified the sensor as, this is something where if you knock it out, it impairs galvanotaxis, but it doesn't impair cell motility. And it has the properties exactly what you think it should. It's very highly charged, it's a membrane protein, and it can couple to the cell's polarity. So we think this is a really strong candidate. So let me show you this movie. The sensor, they've named it galvanin. So here's a fluorescently labeled galvanin within HL60. You turn on the electric field, you see the sensor comes to the back, and then the cell responds. See that again, the field turns on, sensor goes to the back, the cell responds. So this lets us quantify the anisotropy directly, because you can just measure the fluorescence directly. So you can measure the anisotropy by looking at the ratio of the cathode to anode fluorescence. And you see that when the electric field is off, it's uniform as you'd expect. And when you turn the electric field on, you see a change in the anisotropy. And that's the experiment, and that's our fit to theory. But now we said that the time scale to respond is exactly the same as the time scale for the anisotropy to forget. So if you turn the field off, we can predict what should happen just by our fit to the initial data. So you turn the field off, and you predict what happens, and you get this beautiful fit predicting what the anisotropy does. And we can go further. We can also measure the directionality. That's the direction that the cell is actually going. And you can see that it turns on when the field turns on and forgets exactly like we predict when the field turns off. So I was really excited about this because also this is like a simple enough model. We can now constrain all of the parameters in the model. You can see that, measuring the degree to which this anisotropy shows up tells us what the Peclet number has to be. It turns out to be -0.5. you might have noticed we got the wrong sign for some of the earlier things. It doesn't actually matter. The results are the same, independent of the sign. But we're in this regime where linear response works, so that's good. You find the time scale for response. It's actually quite fast. It's 35 seconds. They can even measure the diffusion coefficient of this protein. We can see that it's consistent with just diffusion. You have to be a little bit careful about the cell geometry to do that, though. So this completely characterizes the response of the anisotropy, this cathode to anode ratio, but it doesn't characterize the response of the directionality. There's one additional fit parameter to characterize the response of the directionality, and it's the number of sensors on the surface of the cell. In principle now, we could fit this and predict, here's how many galvanin are on the surface of the cell. So we did that, and here's the number we get out. The number is 4. And so we know that's not actually the right number of galvanin on the surface. So what this tells us is the stochasticity in this problem is not just the randomness of the sensor locations. It tells us that there has to be additional noise in service of that. Or another possibility is that essentially the galvanin are really highly correlated. And so there essentially are only 4 independent protrusions at any given time. So we're trying to think about what this means in terms of modeling this, but right now we think that it's just indicating that there's additional noise that overwhelms the stochasticity from the individual sensor shot noise. And then we can generalize things to things like pulsed fields. People care a lot about pulsed fields for medical applications, because if you're going to design like an electric field bandage or something like that, you don't want to use DC fields because you can end up with some weird electrochemistry happening. So people have tried using pulsed fields. It turns out it doesn't buy you a ton. If you pulse the field at 50%, it's like having the field on at 50% strength. Okay. So let me wrap up. We said that cells can make this simple calculation to estimate the field direction. We can collapse a lot of different cell types onto one universal curve. And we have this linear response theory that characterizes response to dynamic fields. I also should thank all of the support. And, just since I'm not going to have time to talk about it, I want to show the few cartoons and movies from other things the group does. So thank you very much for your time.